Welcome back, everyone. Today, we're going to be doing a deep dive into a research paper about how muscles work after a dog has had surgery for intervertebral disc disease, IVDD.
Yeah.
So this research uses a really cool technique to try and understand this.
I see.
It's not just about, can these dogs walk again? It's about how are their muscles firing compared to healthy dogs?
Yeah. Yeah. This is a really, it highlights a lot of the complexities of neurology and how much we still have to uncover about the recovery process.
And, IVDD, intervertebral disc disease, is a very common spinal problem.
Absolutely.
In dogs.
Especially
those adorable long-bodied braids.
Yes. The dachshunds. Yeah.
And typically, vets use what's called the modified Frankel scoring system.
Yes.
To assess, how a dog is doing after
their mobility,
IVDD surgery.
After surgery.
And that's helpful.
It is, yeah.
But it doesn't tell the whole story.
It doesn't tell the whole story.
It's like, categorizing books by genre.
Okay.
Like you get a general idea, but it doesn't tell you. The plot or the character.
Yeah.
And this is where this study comes in.
Okay.
Using a technique called SEMG.
Right.
Which stands for surface electromyography.
Yes.
To get a much more detailed picture.
Much more in depth.
Of muscle activity.
And SEMG is actually very well established.
Okay.
In human spinal cord injury rehabilitation.
Interesting.
It's surprisingly underutilized in veterinary medicine.
Huh.
So this study is really pushing the boundaries of what we can understand about muscle function.
So how does this SEMG work? Is it like an X-ray for muscles?
It's more like listening to your muscles talk.
Interesting.
It measures the electrical activity in muscles as they're contracting and relaxing. So it gives a really precise picture of how those muscles are working.
Yeah.
And they focus on two specific muscles in this study.
Okay.
One is called the vastus lateralis.
Okay.
It's in the thigh.
Gotcha.
And the other is called the gluteus medius.
Okay.
And that's in the hip.
Yeah, so those are some big, important muscles for locomotion.
Yes.
And they had 7 dogs.
Right.
1 month after they had spinal surgery.
Yes.
And compared their muscle activity to a control group of 7 healthy dogs. So they have their 2 groups of dogs.
Yes.
All carefully selected and matched. And
making sure they fit the criteria.
No other health problems or anything like that.
Exactly, yeah.
And what did they actually have these dogs do? Yeah.
So it was pretty straightforward. They just had them walk on a non-slip surface while the SEMG was recording their muscle activity.
So just a normal stroll.
Just a normal stroll, yeah.
Nothing too strenuous.
Yeah, nothing crazy.
I bet those dogs were thrilled to be part of this.
Yeah, I'm sure.
Groundbreaking research.
Yeah.
So what did the SEMG reveal about their muscle activity?
So there were some unexpected findings. They actually found significantly greater muscle activity in the dogs who had the spinal surgery.
Wait, so
compared to the healthy dogs?
The dogs who were recovering from surgery had more muscle activity.
More muscle activity, yes.
It seems counterintuitive.
It does, yeah.
Shouldn't their muscles be weaker?
You would think, wouldn't you?
Yeah.
But here's where it gets interesting. IVDD often leads to something called
upper motor neuron syndrome.
Upper motor neuron syndrome.
Or UMN syndrome for short.
UMN syndrome.
Yeah.
That sounds serious.
It can be.
Okay.
And it's all about how the nervous system controls muscles. So when the spinal cord is compressed, it disrupts the signals that travel from the brain to the muscle.
To tell the muscles what to do.
To tell the muscles what to do, exactly.
Yeah.
And these signals normally inhibit muscle activity.
Oh, okay.
They act like brakes.
Gotcha.
But when those signals are damaged, you get overactive muscles.
Okay.
It's not that the muscles themselves are stronger.
Okay.
It's that they're firing more than they should be.
So it's like the volume control is stuck on high.
On high.
Gotcha. Yeah. That makes sense.
Yeah.
So it's not just about muscle weakness.
It's about this complex interplay of nerve signals. And the body trying to figure out.
The body trying to move.
Compensate. Yeah.
That's fascinating.
Absolutely. And speaking of complexity.
Okay.
The researchers uncovered another interesting finding muscle activity was actually lower on the surgical side in the post-op group.
Now that is a head scratcher.
Yeah.
Why would that be?
Well, if we think about the bigger picture, it's important to remember that the surgical side is usually the more affected side to begin with.
Got you.
So you have this interplay of factors.
Yeah.
The surgery relieved the compression.
Right.
Which is good.
But
there's also the potential for some nerve damage during the procedure.
Gotcha.
And then on top of that, the dog's body is trying to compensate.
Figure out new ways to move. Exactly.
Yeah, it's like a dance between healing potential nerve issues and the brain rewiring things.
Exactly.
And that's really cool.
Yeah.
And the study also highlighted this fascinating parallel to what we see in humans with spinal cord injuries.
You mean they're seeing similar patterns?
Yes.
Of increased muscle activity?
Research using SEMG in humans with spinal cord injuries has also shown increased resting muscle activity.
Gotcha.
Mirroring what this...
So it's the same kind of phenomenon.
Very similar,
Wow.
Which really strengthens the idea that similar mechanisms might be at work.
That's incredible.
Yeah. It's fascinating.
So what does all of this mean for our furry friends recovering from IVD surgery?
Yeah, what are the implications of these findings?
That's the burning question,
So if we can better understand the underlying reasons for these unusual muscle activity patterns, we can potentially develop more targeted
and effective rehab therapy,
rehabilitation therapy.
Absolutely.
That's not just about getting those legs moving again. It's about understanding the why behind the movement.
Yes.
I'm sensing a but coming.
Of course,
Yeah.
Like any research, there are limitations.
So let's dive into those limitations.
Sure.
What are some of the challenges that the researchers encountered?
So one of the key challenges was the small sample size.
Right.
They only had 7 dogs in each group, which is understandable given the specialized nature of the study.
Sure.
But it does limit how much we can generalize the findings
to other dogs.
It makes sense.
And then also working with these small breeds, like dachshunds can be tricky. Getting those SEMG sensors on their tiny legs, it's no easy feat.
Yeah, those are some little bitty dogs.
They are.
Yeah.
And then there's the issue of comparing these findings to human research.
Right, because you can't just ask a dog to flex as hard as you can.
Exactly.
Great.
So in human SEMG studies, they normalize the data to a person's maximum voluntary contraction.
Gotcha.
Which gives you a clearer picture of how much effort they're putting in.
Sure. So you have that baseline to compare it to.
Exactly.
But with dogs, they have to use...
You have to use different methods?
Different methods to analyze the muscle activity.
To analyze it.
So there's a need for more...
more standard protocols.
Yeah.
For using SEMG.
Yeah.
In veterinary medicine.
In vet med, absolutely. More research with larger sample sizes and a wider range of breeds would be really valuable too.
Yeah, to make sure this...
To make sure that it's...
Applies to all dogs.
Applies to everybody,
Not just the dachshunds.
Yeah, not just the dachshunds. Okay. But as this technology becomes more widely used, I imagine we'll start to see, more refined
techniques.
Techniques and protocols and that sort of thing.
Absolutely. It's very exciting to think about the possibilities.
It really is. I mean, imagine being able to use SEMG to, predict how well a dog might recover after surgery.
Yes, how well they'll do.
Or to tailor rehab plans.
Yeah, to tailor their rehab plan.
Yes,
based on their specific...
That individual dog's muscle.
Yeah, their muscle activity patterns. Activity patterns?
Exactly.
That would be amazing.
It would be incredible.
So instead of relying solely on the modified Frankel score, which we know has its limitations.
Which has limitations, yes.
Vets could use this to really...
To really zoom in. And see what's happening.
Yeah, on a much
deeper level. Yeah.
That's cool.
Absolutely.
So are there any tools or techniques from human medicine that we could adapt? For dogs?
Sure. There are a few promising avenues to explore.
Okay.
One is called the Ashworth scale.
Ashworth scale?
It's used to assess muscle spasticity in humans with neurological conditions.
So it's basically a way to measure
how
stiff or tight a muscle feels.
How stiff or tight a muscle feels, yeah.
Okay.
And it involves passively moving a limb and just feeling for resistance.
Interesting. And then there's
the Olby scale.
The Olby scale.
Yeah. Which is particularly interesting because it was specifically developed for dogs.
For dogs.
With spinal cord injuries.
Spinal cord injuries, yes.
So instead of just saying, this dog has a grade 2.
A grade 2.
On the modified Frankel score.
On the modified Frankel score, yeah.
Vets could use these scales.
You can use these scales to get a more
nuanced and detailed understanding of how they're recovering.
Yeah. That's great.
Absolutely.
So it paints a clearer picture.
It does, yeah.
So it feels like we're on the cusp of...
Does feel like it.
Like a new era?
A new era, yeah.
In canine neurology and rehab.
In canine neuro and rehab, absolutely.
This research and the technology...
the technology.
It utilizes... Could really change the game.
Absolutely.
In how we assess and treat dogs with spinal cord injuries.
Yeah, it's all about
improving
quality of life.
Quality of life for those dogs.
For those dogs.
Yeah, and
who knows?
As more research is done,
maybe these findings in dogs could even lead to advancements.
Yes.
In human spinal cord injury research.
Absolutely.
The connections between animal and human health.
It's fascinating.
They're always fascinating.
Yes.
And I think what's so powerful about this research is
that really?
It underscores
the
importance of
understanding the why.
The why.
Behind the movement. Or lack thereof. Yeah, that's the crux of the matter.
It's not enough to just observe.
It's not,
that a dog is struggling to walk.
Right. We need to understand.
You need to delve deeper. And understand those underlying,
neurological and muscular factors.
Yeah.
Neurological and muscular factors that are at play.
That are going on.
And that's where SEMG comes in.
It
provides that window. Into
that intricate world of muscle activity
and nerve
signals. Yeah,
it's really quite remarkable.
It is, yeah.
So to bring this all back to our listener, what would you say?
What would I say
is the key takeaway?
The key takeaway is that
from this deep dive,
recovery from spinal surgery, whether it's for a human or a dog, is a complex process.
There's a lot happening
under the surface.
And the modified Frankel score, while it's a helpful tool, it only tells part of the story. Yeah.
So this research
highlights the potential of that solution.
Revolutionize
the way we assess and treat.
Yeah, it's about moving beyond
spinal cord injury.
Those broad generalizations and embracing the individuality of each patient.
It's personalized medicine.
It is, whether they have 2 legs or 4.
2 legs or 4. I love it.
Yeah.
This research opens up.
It really does.
So many possibilities.
Yeah.
For the future.
For the future of
veterinary neurology.
Yeah.
Imagine a world where we can create truly personalized rehab plans.
Yes. Tailored to
based on
their individual needs.
A deep understanding of each dog's unique needs.
Needs and recovery patterns.
Exactly. That would be incredible.
It would be amazing.
It's a future worth striving for.
It is.
And with continued research.
And
technological advancement.
And tech advancements.
I think we're well on our way.
I think we are too.
This has been
great.
Such an insightful exploration of SMG. And its potential to change the landscape.
To really change things.
Of canine spinal surgery recovery.
Absolutely.
It's amazing to see how technology like this is giving us
such a good understanding.
Such a detailed understanding.
Yeah, of what's happening.
Of muscle activity.
Yeah.
And how it relates to spinal cord recovery in dogs.
It's a reminder that even though we're talking about really complex science, the ultimate goal is simple.
Yeah.
It's to help these dogs.
Help the dogs.
Regain their mobility and enjoy a good quality of life.
Exactly.
And it's really a testament to the power of scientific curiosity and collaboration.
Yes.
I mean, this research wouldn't be possible without the dedication of veterinarians and researchers. And, of course, the canine participants themselves.
Of course. It's a beautiful thing to witness.
It is.
So To our listener, we'll leave you with a final thought-provoking
question.
Question.
Okay. Could this be the key to unlocking even more personalized and effective treatments for dogs after spinal surgery? Could it help us move beyond those general assessments and create rehab plans that are truly tailored to each individual dog's needs?
It's a question worth pondering for sure. And as this field continues to evolve, I have no doubt that we'll continue to see more and more exciting breakthroughs that benefit our furry companions. This has been a really illuminating deep dive. It's amazing to see how technology like SEMG is giving us this detailed understanding of muscle activity and how it all relates to spinal cord recovery in dogs.
It is. It's a good reminder that even though we're talking about, really complex science, the ultimate goal is simple. Help these dogs regain their mobility and enjoy a good quality of life.
Exactly. And it's a testament to the power of,
scientific curiosity.
Scientific curiosity and collaboration.
And collaboration. This research wouldn't be possible without, the dedication of veterinarians and researchers and, of course, the canine participants themselves.
Of course. It's a beautiful thing to witness. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.